Hi, I'm Kayla, and today we'll scratch the surface on some basics of microbiology. So a classic microbiology fun fact is that your body is home to more microbial cells than human cells, which if you didn't know, you're welcome, and it's something fun to ponder. It makes me kind of wonder if we're more microbe than we are human. But if that didn't get you, then just contemplate that a teaspoon of soil has 1 billion microbes in it. Microbes are everywhere. They are arguably the main colonizers of planet Earth. However, to the general public, they often get a bad reputation, but the world of microbiology is so much larger than just the pathogens or microbes that get us sick. So today we're going to sample the human body and use that as a jumping off point to learn about the microbes that we find there. Specifically, we're going to focus on 3 things. First, the type of microbes found all around us. 2, how microbes differ in their morphology, which is a science word for form and structure of something. And 3, we're going to start floating the idea that microbes are each adapted to their own ecological niche. Basically, think of your skin, your mouth, the soil outside, each as its own microenvironment with varying environmental conditions. That means that the microbes that we find there are all going to be well suited to live in those conditions. All right, so we've got some petri dishes, and we're going to culture microbes. Culturing microbes basically means intentionally grow them. Inside of the petri dish, we have culture media, which is essentially microbe food. It has nutrients that are going to target the microbes you want to grow, and it also has agar, which is a solidifying agent. In this case, it's extracted from seaweed. We're going to use tryptic soy agar, TSA. This is a standard culture media. Lots of microbes are happy growing on here. We'll also use some sterile swabs. So these swabs have been autoclaved, meaning they've basically been pressurized at high temperatures, and everything inside of this vial should be sterile. So these swabs will help us sample our environment. First things first, we always label our cultures with the important information, like your initials, date, and identification of the microbe or sample. For example, mouth is sufficient for the mouth culture. Pro tip, always label the bottom of the petri dish, the part that actually holds the media. That way, there's always a label attached to your culture. If you label the lids, for example, there's a chance that things could get swapped just due to human error. We also incubate upside down, which is maybe not intuitive at first, but it helps prevent condensation and dripping onto your sample. I've already given you a sneak preview that I've sampled my mouth and my armpit. I also sampled my hand by simply touching it to the petri dish, and then took a quick trip to get a soil sample from the topsoil right outside of the science building. Next, we toss it all in the incubator. Warmer temperatures help encourage microbial growth and generally speed things up. Too high of a temperature could cook our microbes, but 37 degrees Celsius is a great temperature if we're trying to culture microbes from the human body. What if I told you that 37 degrees Celsius is about 98.6 degrees Fahrenheit? Yep, that's human body temperature. They'll stay in the incubator for about 48 hours. Just note that incubation times will vary depending on your goal and what microbes you're trying to culture. For our purposes, that's the incubation sweet spot. So once we've hit that mark, they're transferred to the refrigerator, which will slow down growth and preserve the samples for a few days or just until we're ready to look at them. What I immediately notice are differences in colony morphology. That's referring to differences in the characteristics, visible form and structure of a colony on the plate. So each colony is actually not just one microbe. Microbes are super, super tiny, and we would need at least a compound microscope to take a look at them. So a colony is then what was likely one bacterium that landed on this plate. And then because bacteria grow by binary fission, which is asexual reproduction, they basically just replicate themselves. One microbe becomes 2, which becomes 4, which becomes 8, and so on until you get this big visible mass of growth that we can see on a petri dish. And because they divide asexually, we would expect that all of the microbes inside of a colony are genetically identical if we're ignoring mutations. Colony morphology includes shape, like circular or irregular pattern. It could refer to texture, like smooth, rough, or wrinkled. For example, a lot of the growth on our soil plate is really wrinkly in texture. And it can even refer to color. Some of these are yellow and some of these are white. So these characteristics really help us in figuring out what microbial species might be on this plate even before we bust out a microscope or do any physiological tests. By the way, let's point out a common point of confusion. So cellular morphology and colony morphology are not the same thing. We know now that colony morphology is referring to the growth of many microbes in a lump on a petri dish, whereas cellular morphology is talking about the form and structure of a single cell of a microbe, be it a bacteria or a fungi. Now it's time to prepare some slides so that we can take a look at these microbes under the microscope. Once again, we start by getting organized and labeling our slides. I add 1 drop of water to the slides so that when I grab some of the microbes off of the petri dish, they can then be suspended in this water droplet. And then I'll spread out the water droplets so that we have an even surface and a faster drying time. In this clip though, you'll notice I've placed 3 drops of DI water onto my slide. That's because I want to view 3 different colonies separately from the same hand sample. But I want to have them all on the same slide for easier viewing under the microscope. You might also notice that I flamed my inoculation loop before dipping it into the petri dish to grab some of my culture. So placing the inoculation loop into the hottest part of the Bunsen burner until it turns bright red ensures that the loop is sterilized and won't contaminate my sample. The next steps are going to be heat fixing the microbes onto the glass slide. You can do this by passing it through the Bunsen burner a few times or by using a slide warmer, which in this case I did. And then we need to stain the slide for easier viewing under the microscope. Let's circle back to the human skin as an environment for microbes. So it's actually a pretty extreme environment. It's dry, there's constantly dead skin cells sloughing off, and it's salty. I mean, think about a time that you've ever done a really hard workout, or maybe it's just a really hot day and you're sweating a lot. As you sweat, salt starts to accumulate on your skin. So anything that lives on your skin has to be somewhat salt tolerant. So just to place everything in context, the whole tree of life is divided into 3 domains: bacteria, Archaea, and Eukaryote. And surprisingly, or maybe not surprisingly at this point, it's pretty much dominated by microbes. By the way, the people who study how things are classified are called taxonomists. They basically work to take organisms and fit them into these different grouping categories. Taxonomy, or this tree of life, is always being revised as we develop new technologies and just generally have more research, but all organisms can fit into a grouping. So the grouping starts really broad and it keeps branching as we get into groups that are smaller and more specific. So organisms get their scientific name or Latin name based off of this classification system. The 2 lowest levels of the grouping system are genus and then species, although there's some exceptions for the microbes, but we'll keep it at that for now. One of the most common skin microbes and really well studied is Staphylococcus epidermidis, and this is going to fall under the domain bacteria. Staphylococcus refers to the genus name, and there are many different species of Staphylococcus, but epidermidis refers to that one species that we'll be focusing on that lives on human skin. So to really drive this home, I like to think about it as Staphylococcus, or the genus, being like your family's last name. So my name's Kayla Delventhal, right? And Delventhal means that there's others in my immediate family. There's my dad, Dan Delventhal, and my mom, Cynthia Delventhal. But I'm the only one named Kayla. So similarly, Staphylococcus epidermidis has many other Staphylococcus relatives, but it is the only one that is epidermidis. It is that species. Staphylococcus epidermidis has a really unique cellular morphology. It's actually why it has the name Staphylococcus. So staph has an origin in Greek for clustering, referring to a clustering of grapes. And coccus refers to a spherical shape, coming from the Greek word kokos, meaning berry. Staphylococcus epidermidis is adapted to this dry, salty environment of our skin. And it's actually one of our mutualistic symbionts, meaning we give a nice environment and it gives us a little something something in return. So fun fact, for an example, out of the numerous studies on this microbe show that it can actually stimulate keratinocytes, so these cells on the layer of the epidermidis, to release antimicrobial peptides that are going to keep pathogens and acne-causing bacteria at bay. So thank you, Staph. Nevertheless, under the wrong conditions, like if Staphylococcus epidermidis were to somehow get into your bloodstream, it can cause an infection. And this really brings home the point that context is so important when considering microbes. Let's come back to the first part of the digestive tract, the mouth. So remember Staphylococcus? We're going to meet another coccus-shaped bacterium, Streptococcus. Just like Staphylococcus gets its name from its staph arrangement, or its bunching, and its circular shape, coccus, Streptococcus has been named Streptococcus because it is circular in shape as well, but instead it forms chains. Once again, the prefix strep comes from the Greek word streptos, meaning twisted or twined, and it's referring to this bacteria's ability to form chains. So what makes these coccus-shaped bacteria form chains instead of bunches like our Staphylococcus? Well, it all has to do with their plane of division when they do binary fission. This is asexual reproduction, and to oversimplify it, they basically double their cellular contents and then chop in half. If the coccus-shaped bacterium divides along the same plane of division every time it does binary fission, then you can end up with a chaining pattern. If that plane of division changes every time the bacterium does binary fission, then you can end up with bunches or even something like tetrads. Have you ever had strep throat? Because I bet now you can guess who the culprit is. In that case, it would be Streptococcus pyogenes. But not all Streptococcus species are pathogenic or disease-causing for us. We actually have a ton of Streptococcus species in our mouth. I bet I'd find a ton on this plate if we check it out under the microscope. They really love this wet, nutrient-rich environment. Interesting. So we did a gram stain on this stuff in my mouth, and this is cool. I'm seeing some really obvious Streptococcus microbes, and then it looks like some of the yeast got cut up in that gram stain because we have some irregular patterned little dudes. Okay, this is fun. Oh, and some bacillus-shaped microbes. We got a little bit of everything going on here. Moving on, I want to talk about the digestive tract. I'm definitely making a stretch here because we only sampled my mouth environment. I couldn't feasibly sample my digestive tract for us here today, but I still want to talk about it. So different sections of the human digestive tract, though all connected, contain distinct microenvironments. You're probably seeing a theme here. For example, closer to the mouth, we're going to see a higher percentage of oxygen availability versus the colon, where many species are even anaerobic, meaning they don't use oxygen at all for their cellular respiration. And even some of these species would just die on contact with oxygen. Closer to the stomach, we're going to have a really low pH, so a very acidic pH. And then as you move away from the stomach through the small intestine down to the large intestine, we're generally seeing it become more alkaline. Two of the most common groupings of bacteria in the digestive tract are the Bacteroides and the Firmicutes. This is referring to the phylum level, which is pretty high up there on that taxonomic system that we talked about. We've learned that bacteria can be circular in shape when we looked at Staphylococcus, but there can be other morphologies that we see when we look under the microscope. For example, another very common cellular morphology for bacteria is bacillus, meaning rod-shaped. This actually comes from 19th century Latin, meaning stick. So for example, you can find many species of lactobacillus in the small and large intestine. Lactobacillus is a bacillus, rod-shaped bacterium. And if you check the label of your yogurt that you buy from the grocery store, you'll actually probably see lots of these species added into yogurt as a probiotic. It also gives the yogurt that tangy flavor thanks to the fermentation capabilities of our lactobacillus. By the way, remember how we got a soil sample from right outside of the building? Well, I expected to find a lot of bacillus-shaped bacteria in this sample, and especially when I was looking at the petri dish and I saw some of those wrinkly colonies, I was suspicious. And sure enough, when we look under the microscope, we see lots of diversity and lots of bacillus shapes. Now, I've got to mention the Archaea. The Archaea are another domain of life, one of those big groupings that we talked about. It's the highest level on our taxonomic tree. There's not nearly as much research and understanding on the Archaea as there is bacteria, and that's mainly because they thrive in really extreme environments. Think sulfuric hot springs or deep sea hydrothermal vents. And because of that, they can also be kind of hard to just culture up on a petri dish like we can with bacteria. On top of that, there are no known human diseases that are directly caused by Archaea. They can be implicated in a dysbiosis, which is like an imbalance of the human microbiome. More on that later. But because they don't directly cause human disease, you can understand why humans historically haven't spent that much time and resources on studying them relative to the bacteria. Similar to the bacteria, Archaea have a wide range of morphologies, often resembling bacteria. For example, spherical coccus shape or the rod-shaped bacillus, and even spirals. Actually, for a long time, because their structures are pretty similar, taxonomists thought that they were all the same, the bacteria and the archaea. They were called the prokaryotes, which is a word that we're now kind of trying to phase out of microbiology because it's pretty misleading. Because while they have structural similarities, the archaea are really different from bacteria in terms of having a really diverse cell wall, diverse cell membranes. And that's not even mentioning their ecology and metabolism and evolutionary history. Now at this point, we've talked about the bacteria and the archaea, but what about the eukaryotes? Well, before we do, I want to clarify the big identifiers of eukaryotic cells that make them different from the bacteria and archaea, or the prokaryotic cell types. All cells will have a plasma membrane, but eukaryotes will also have a membrane-bound nucleus, where the genetic material is housed. Bacteria and archaea, on the other hand, have a nucleoid region, but it's not bound by a membrane. Similarly, eukaryotic cells will have membrane-bound organelles, like the mitochondria, which has many functions, one of them being the powerhouse of the cell. It's important for energy production. Side note, here's a biology puzzle. Bacteria also need to produce ATP, the energy currency of the cell, but they don't have a mitochondria, so where do they do it? See if you can figure it out, and we'll answer that one in another video. So did we find any of them on our sampling plates? Well, if you see any fuzzy growth or filamentous growth, that's a telltale sign that you're looking at a fungi. Fungi can definitely be macroscopic. But maybe what not everybody knows is that fungi can also be microscopic, and they're generally divided into two categories. We have the yeasts and the molds. Molds are the fuzzy ones. They're really morphologically diverse, but generally speaking, they have hyphae, which is these filamentous cells. They kind of remind me of like roots of a tree. And they also have specialized structures that contain spores for reproduction. That's why they often look fuzzy when they're growing on a petri dish. That fuzziness would be those stalks with spores on the end, creating this 3D fuzzy structure. You've probably seen this if you've ever let a piece of bread get old and moldy. It's got that maybe green, dark brown fuzz to it. There's also the single-celled fungi, the yeasts. They divide by a really cool process called budding. It's basically a funky form of mitosis. The new cell will start as a bud on the mother cell, and it will basically keep growing until it has all of the cellular components it needs, including genetic material, and then it will eventually detach from the mother cell. Speaking of yeast, when I first looked at my armpit culture, I wasn't really sure what was going on there and kind of excited to look at it. And sure enough, when I look under the microscope, I see what appears to be a yeast. We have these irregular circles, and I even see some evidence of budding. Candida albicans is a great example of a yeast that's found in many different microenvironments on the human body. Candida can sometimes get a bad reputation, though. For example, let's say that you need to take antibiotics to attack an infection. By the way, antibiotics specifically target bacteria. So what might happen is you have a large die-off of bacteria in your body, but then you have these candida yeasts that are just sort of looking around and everyone's gone, and suddenly they have all this space and resources, and so they just naturally start to divide and multiply. So if it starts to increase in number, that can cause something like oral thrush. Not great, or even a vaginal yeast infection. Yeasts are also super important helpers in creating some of our favorite foods. So if you're someone who enjoys sourdough bread or beer, you can thank a lot of yeast microbes for their services in fermentation. So we've talked about the fungi. Are there other eukaryotic microbes? Yes, definitely, but I admit that this part of taxonomy is really tricky to talk about, and that's because the eukaryotic groupings of microbes are constantly being reclassified. Why? Well, you have to remember that DNA and DNA sequencing technologies are actually relatively new, but they're only becoming more advanced and more accurate and more available to people. So as we gain new information, we now understand that a lot of our old groupings were what we call polyphyletic. That means that the group doesn't necessarily share a common ancestor. So for an analogy, something that's not polyphyletic would be the canines. We know that this group, which includes wolves, coyotes, and dogs, are more closely related to each other than they would be to, say, the felines or the cats. On the other hand, our microbial group, the Protists, are from different branches of the tree of life. They don't share a common ancestor in the same way that the canines do, so they're considered polyphyletic. The Protist group, which is sort of being phased out because it's polyphyletic, includes microbes like the algae and the protozoa, which themselves are also polyphyletic. I like to think of the protozoa as mini animals. They include cool organisms like amoebas or the parasite that causes giardia. By the way, definitely do a little Google search on that one. They look like a mixture between aliens and those sucker fish that you find on the side of sharks. And that's also because they actually sucker onto your intestinal lining, like the parasites that they are. And the algae, surprise, they're actually not plants like many people think. They are their own separate category of eukaryotic microorganisms. So bringing it back to our exploration of the human body, I've got some food for thought for you. What happens to the microbial communities on your body when the environment changes? Say you go from a carnivorous or omnivore diet to a vegan diet, or you drastically cut out your sugar intake. Would you expect to see changes in your microbial community? It's actually more complicated than you'd expect. And would you expect that change to be big or small, if any at all? You can see that the microbial world is extremely diverse. We just scratched the surface, literally and figuratively. So thank you for joining me on this journey to an introduction to microbiology. Before we go, let's recap the 3 big main ideas that we just explored together in this video. First, we met the microbes across the tree of life and learned that there are 3 domains of life: the eukaryotes, bacteria, and archaea. Domain is the highest level of our taxonomic system, which branches into more and more specific groupings all the way down to the species level. And actually, many bacteria are even classified into subspecies categories called strains. We also discussed colony morphology by examining macroscopic colonies on a petri dish when we sampled different parts of the body and soil from right outside. We made an important distinction between colony morphology and cellular morphology, which describes cellular structure and form for a given species. For an example, bacteria can be circular or coccus-shaped, bacillus or rod-shaped, and more, and can exist as individual free cells or found in arrangements like chains or bunches. We also began to explore the idea that microbes are unique to certain ecological niches. For example, a microbe found in the mouth where oxygen is ample likely won't survive or thrive in the lower areas of the digestive tract, like the colon, where oxygen levels are really low. Now, as a final fun thought, I invite you to reflect on who's missing. There's a group of kind of microbes that are a huge reality of human life. Can't think of it? Just think back a few years to the COVID-19 pandemic. I'm talking about the viruses. The viruses aren't even mapped onto the tree of life. Why do you think that is? Think on it and we'll explore it later.